So I presented data from our first-in-human phase 1/2A study of the first-in-class next-generation CDK4 selective inhibitor, PF-07220060, in combination with endocrine therapy in patients with HR-positive, HER2-negative metastatic breast cancer who progressed on prior CDK4/6 inhibitors. I presented data from cohort 1B, which is a combination of the CDK4 selective inhibitor together with letrozole, and also cohort 1C, which is the combination with fulvestrant. This compound is a highly selective and potent CDK4 next-generation inhibitor with significant sparing against CDK6. During this cohort, we enrolled patients at doses of 300 and 400 milligrams twice daily. All patients had previously received CDK4/6 inhibitors, and the majority of patients had also received prior chemotherapy agents and other endocrine therapy as well. This was a heavily pretreated patient population with a median of 4 prior lines of therapy in the systemic setting. This drug was found to be well tolerated with mainly grade 1 to 2 toxicity, with neutropenia, as well as GI and fatigue as toxicities. Grade 3 or worse toxicities were rare, and there were very few dose reductions or drug discontinuations. In terms of efficacy, we observe complete and partial responses by RECIST criteria in 32% of patients. The actual disease control rate was 81%, and the median progression-free survival was 8.1 months. The clinical benefit rate was approximately 64% in patients as well. And these were in patients who had previously received prior CDK4/6 inhibitors. In addition to that as well, interestingly, we observed responses in patients who had ESR1 mutations, as well as in those who had PI3K pathway alterations like PIK3CA, AKT, and PTEN mutations. So to conclude, PF-07220060 is a potent CDK4 selective inhibitor with significant sparing of CDK6, favorable safety and tolerability profile with low hematologic toxicity, mainly grade 1 and 2, and infrequent dose reductions and drug discontinuations were observed in this very heavily pretreated patient population with HR-positive, HER2-negative metastatic breast cancer who progressed on prior CDK4/6 inhibitors and endocrine therapy. Importantly, encouraging clinical activity was demonstrated irrespective of ESR1 and PI3K pathway mutations, such as PIK3CA, PTEN, and AKT mutations. These data strongly support the ongoing phase 3 open-label randomized clinical trial evaluating PF-07220060 plus fulvestrant versus investigators' choice of therapy in patients with HR-positive, HER2-negative advanced metastatic breast cancer who progressed on prior CDK4/6 inhibitor-based therapy.